Good. Good that you're good, Courtney. Anybody do anything exciting this weekend? Other than watch the Lions lose another one? Should get used to that by now. You bought a house. Well, you got me, Jessica. I bought a repaired watch. That's about the extent of my weekend. Excited about your house, or got your rooms all planned out? Now you just get the joy of spending every weekend fixing it. Yes. Well, good luck. It is exciting. It is exciting. I only bought a house 2 years ago. Yes. Many days I wish I did not. I like someone else to come and fix things when they break. All right, there's 12 of us here today. We'll give a minute to anyone else who wants to walk in. I'm going to give you a little review on stats. If you want to pull out the PDF that I uploaded, I'm going to show you 6 or 7 slides and just review those pages. You're welcome to take notes. I don't think I posted those slides. If you think those slides will be useful, I can post them after our session this morning. And then I will spend about 15 or 20 minutes showing you Excel. If you're an expert on this, then you can give me the pointers. But since we're going to use Excel to perform many of these basic stats, this is what we're going to start with. Okay? So if you want to download that PDF and get it open, you can follow along. I'm not going to follow it word by word. I'm going to do my own thing to give you a different version of what is written. And if you want to open up an Excel file and just put some numbers in there in a column, I don't care. I'm going to show you some tricks and how to navigate, and then you can take notes on that. All right? Questions for me before we begin? Any questions from last week or anything we've discussed to date that you would like to ask? Okay, I'm going to share my screen. It is going to be a PowerPoint that should show up. I can make it full window, full screen. Be aware when I do this, I will not be able to see the chat box, okay? So you can tell me, is that big enough for you to see, or would you like to make it full screen? I need 2 people to reply. That's good. Okay. All right. Let's see if I can get rid of this crap. Okay, better. Okay. So that little manual, that little appendix I gave you, so I can still see the chat box if you have a question. That little manual I gave you, I'm going to back up and actually say, why do we need to evaluate our data sets using statistics? Why? Why are you asked to take a statistics course as a biology major? So the big picture is you have these data points that you collected, okay? So the vocabulary here is data is plural. Datum is singular if we talk about a single point. So I usually avoid this problem of data versus datum by referring to as a data matrix or a data set, right? So those will be singular. And obviously data sets is plural, and data matrices are also plural, okay? So that's going to be my vocabulary. So I'll use data point a lot. All right, so you've collected these data points. The data points came from your observations from your experimental design. And so at the end, you want to make a statement, right? You want to make that inference. Well, it's very hard when you start looking at tables of numbers to actually find a pattern. And so the statistics that we're going to use summarize these collection of data points and describe the data, okay? Now, they're going to be different descriptions of the data. Sometimes they're going to tell us what a value is, where the quote unquote center of all the data points are. Sometimes it will tell us how spread out the data points are relative to each other. Sometimes the statistic will tell us how much error there is from the actual or true value. Okay, so they can describe all kinds of elements about your data matrix. The other thing that we're going to use statistics for is the whole goal of stats is to tell you where the error is in your experimental design and to quantify the error. That's number 3. So in other words, quantify, you want to put a value on it. How big is the error? Can I ignore it? Or do I need to look at my values and say, I don't think I can trust anything because the error is so big? Okay, so those are the 3 goals of stats. Now the pictures there represent how you could actually identify where error occurs. So one thing you could do is if you worked in the lab, all right, Jessica, you're working in the lab. Your supervisor gives you 3 different methods to collect the same type of data. And you're going to do each method 3 times. And you're going to calculate the error associated within each method. And so obviously the goal here is to figure out which method produces the most reliable results, the most precise results, and that's the method you're going to go with, right? So you can find errors between different methods. The other thing you can do is have the same method, and if you've got 3 different researchers, so now I got Kelsey, Christine, and Leah in the lab collecting data, right? You want to know, okay, which of you collects the most precise data, which of you collects the least precise data, because how can we improve your data collection, right? So that's a different source of error, right? That's the, what is the user-defined error, okay? So in general, this is what the stats are used for, and I don't think your book really specifies enough, you know, why are you doing this in the first place? This is why, and this is why you're asked to take a stats course as a bio major. Now, this part of the appendix deals only with what are called descriptive statistics. There are other types of stats, okay? But these parameters that we're going to look at, right? So the parameter is going to be the actual thing we're going to calculate, all right? They're going to describe the data matrix, the data set, whatever it is, the table of data points. And we come down to 2 basic questions. Where are the data points along the x-axis? These are called measures of central tendency. Now, the thing in blue is a standard statistical phrase, okay? This is what stats people get all excited about, and this is how they communicate with each other. They talk about measures of central tendency. That's, in English, that's telling us where the data points are along the x-axis. Now, that implies that the data points are continuous data. Okay, so we need to have this conversation first and say data comes in 2 flavors. Okay, there's continuous data, right, which allows for intervals or basically fractions of points. Okay, and then there's discrete data, which are basically categories. So discrete data is, what type of tree is this? Well, that's a beech tree. This is a pine tree. This is a red oak, blah, 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 blah. Okay? There's no such thing as, well, this is a 3/4 pine and a 1/4 oak. That makes no sense, right? Continuous data are things like heights, because you can be 5'8", but you can also be 5'8 1/4 or 5'8 3/8. You get it? So you can have all these fraction of these interval points. So obviously, if we're going to plot it like this and we're going to have a measure of central tendency, these are continuous data, right? The other thing that these stats are going to address is how close are the data points relative to each other, all right? So these are measures of spread or variation, right? And then in each of these columns, I've split it up so it's much more obvious as to what each is going to measure. So the 6 items on the left, really the 5 items on the left, are going to be based on measures of central tendency. They're going to tell you where the distribution is, right? The distribution is all the data points collectively. I'll show you a picture later on, all right? And they're going to tell us something about the x-axis, their location on the x-axis, whereas variation, standard deviation, range, and standard error are going to tell us more about how spread out the data points are, okay? And I'm going to show you pictures of this because it's easier to visualize. So let's start in the left-hand column. I'm going to quickly go through some of these definitions since you've already heard of them before. I'm going to guess, and if you haven't, please ask. All right. So we like to speak of averages all the time. All right. You watch sports. What's the batting average? All right. Really, for the stats people, their hair stands up on the back of their neck. They start to get upset if you use the term average. They use the term arithmetic mean. All right. So if you see the word mean by itself, or if you see arithmetic mean, you can convert that in your head to average. It is the same thing. Now, this is not on the test. We're not going to do this. But why do they call it the arithmetic mean? Because there are other calculations called the harmonic mean and the geometric mean. Okay, and it really depends on what type of data you're collecting, but we're not going to worry about it. So when we calculate the mean, it means the arithmetic mean, the average. And you know the equation for this. This is the sum of all data points divided by the number of data points. And the number of data points is called the sample size, which is number 6. Now, you're going to have to learn some notation because everybody in the world uses this same notation. There's no ways or ifs, ands, or buts about it and to get around it. Okay? So this is the way it is. All right. So that funky symbol, assuming you can see my cursor circling this, this is a Greek letter. It is called sigma, and it is a capital sigma. There is a lowercase sigma. So Greek letters, just like the letters we're used to using, come in capital and lowercase versions, and that is a capital sigma. Okay? May have seen it before, probably seen it on a frat house, a sorority somewhere. All right? That means sum. So whenever you see sigma in mathematical notation, it means sum, which is the fancy way to say addition. Okay, so you are going to sum all the data points and divide by n the sample size. That's probably straightforward, yes? Okay, I'm going to assume that's straightforward unless a bunch of people chime in. The second one is median. And just like the median is in the middle of the highway when you drive down, the median is the middle number in a distribution. You always start on the smallest values on the left side. You line them up or you sort them into the largest value on the right-hand side, and you take the middle value. Now, if there's an odd number of values, that's easy. So if I have a 2, a 5, a 6, a 9, and a 12, that's 5 numbers. I take the third one, which is 6. All right, that is the median. All the median tells you is that there's 2 values smaller than that, and there's 2 values larger than that. You got it? If there are an even number of values, you actually have to take the average of the middle 2 values. So now I'm going to take the average of 6 and 8, which are the third and fourth values, in 6 digits. And so obviously that'll be a 7. Okay? If the distribution had been 2, 5, 6, 6, 9, and 12, then the average of 2 6s is 6. So that's okay. Ready? The mode is the most frequent number in your data set. Should be straightforward. The maximum is the largest, the minimum is the smallest. So all of these tell us about where the data points sit on the x-axis. Variation, the spread is a little more complicated to many students, I think, because the definition looks much more intimidating. But again, if we tear apart the pieces, essentially what you do is you calculate the mean, which you got over here, and then to every value, every data point and observation, you subtract the mean and you square it. Okay? And then you add up all the square differences, and that's called the sum of squares. It's called the sum of squares. Okay? Squares are the deviations. The deviations is a difference, which means you subtract. And again, you're going to divide by n, which is the number of observations, the number of data points. All right? Now, you can put your pencils down. The reason that you have to square it is that you can imagine that there will be some data points smaller than the mean, which would give you a negative number. And there would be some data points that would be larger than the mean that would give you a positive number. And if you add positive numbers with negative numbers, you tend to end up close to 0. All right? And so that's nonsensical in this particular case. So you square it to turn every number into a positive number, because remember, a negative number times a negative number is positive. Okay, so that's why they're squaring it, to get rid of that anomaly if you were just to add negative and positive numbers. All right, the notation for variation is funky S squared. It's italicized S squared. You don't have to italicize it. Standard deviation is just the square root of variation. It's denoted by an S. I understand. I'm just going over the definitions. I'm going to show you visually what it means. The range is the maximum value minus the minimum value, right? And the standard error will come to the last slide. It's going to be the most complicated to understand. So just leave it for right now. Okay? So are there any questions on this? What you need to be able to recognize is the term measures of central tendency, measures of spread, and then which statistic goes under each category. Questions so far? Okay, so we'll go on to the next slide. Now, this is what it means to measure the central tendency. All righty? So if we did, this is called these figures are called distributions. There's an x-axis, there's a y-axis. Because I can graph it as a line or a curve, okay, then that it must be a continuous variable. Leah, no, when I say it's not going to be tested on, when I say this part is not going to, you're going to have to worry about, that's the part you don't have to worry about. You don't have to know a harmonic mean from a geometric mean. I'm just telling you why it's called the arithmetic mean, okay? Does that make sense? But we're going to be using all of these things. So when I go off the deep end about explaining why, those are typically the things you don't have to understand. For me personally, the reason I'm telling you why, I think it's easier to understand if you have the bigger picture, and then you can just ignore the pieces that you don't need. Okay? Plus, I get kind of excited about this stuff. I was a math major. Okay. Even though this is not math. So here's central tendency. I have a distribution. Here's my x-axis. If I can draw a line, that implies that this is a continuous variable. Okay? Everybody talks about a normal distribution, a Bell distribution, right? This is how you typically think of your grades. Well, there's a Bell distribution of grades, right? The grade is a continuous percentage. Low grades down here, high grades up here. Most people fall in this category, all right? This is called a normal distribution because the mean equals the mode equals the median. And so they all fall here in this line. And so when this occurs, that distribution is symmetrical, right? That's a very appealing distribution to look at. You know it's equal on the left and right side. And it is informative, right? It is called a normal distribution. Now, there's other implications to what that means to be normal, but we're not going to go down that road. All righty? Now, this is why you have to use multiple statistics or parameters to think about your data set, because look at this distribution on the right. This is called a bimodal distribution. Bi means two, right? Bicycle means two cycles or two wheels. Bimodal, well, we know what the mode is. The mode is the most frequent value. So now it seems counterintuitive to say there are two most frequent values. That's literally what bimodal means. And so, but you can see it when we look at the graph. There is a large number of these values, whatever it is, and there's a large number of these values, so I end up with two peaks and very little of the other values. Now, this is deceiving, because if I told you that distribution has the same median and the same mean, you might think of this, right? Because it's true over here. They have the same median and the same mean, but so does this distribution. The median is equal to the mean. The only difference is there are two modes, okay? And so at some point, you're probably going to ask yourself, why do I have to calculate all this crap? Can't I just use one value? And the short answer is no, because you would not learn this unless you identified that this particular distribution has two modes, all right? This distribution obviously only has one mode, but these are significantly different. This is often what your grade distribution looks like on a test. These people earned a B and understand it. These people earned a D and did not understand it well. Okay? So from my perspective, I see a lot of bimodal distributions. Okay. Then the distributions can get even wonkier. which is why it's important to calculate all three. So look at these distributions at the bottom. These are called skewed distributions because it's almost like you put your finger here and push it, and the whole thing just blurbs, moves to the left. Or if I put my finger here, the whole thing would move to the right. You don't have to know if this is left skewed or right skewed. Doesn't matter. These are just skewed distributions, meaning they're non-normal distributions. And if you look at the median, so the average here is in green. You see the green line? And then if you look at the mode, the most frequent number, which is in the blue, that's easy to find. That always has to be the top of the peak. And then if you look at the median, which is green, and the mode, sorry, the mean, I keep saying mode, the mean, which is yellow, you'll see that they're not even close to each other. This is a sign, this is an indication that this is a skewed distribution, and this is why you calculate them. Okay, so it's kind of ironic that Jessica bought a house on the weekend, because when you search for houses, the real estate agent often likes to talk about the average value of the house on the street. However, this could be the real distribution of the values of the house on the street. Most of them are down here. There's a few that are up here, and they skew the mean away from the most common or the middle value. Okay? And so it's important when you listen to information in the news that you ask yourself, if they're telling me about averages, is it a normal distribution? Because if it's not a normal distribution, then you get a very skewed, pun intended, a very skewed perspective of the distribution and where the mean sits. Okay? And so what you're going to see in the news, and what you probably, at some point, what you'll see in the news is people complaining about a particular group of people's salary, that it's much too high. And this distribution here is often the distribution of salaries. Most of the people at some corporation do not earn that much money. A few people earn a lot of money. And when you take the average of a distribution, you see the mean. The mean is way over here on the right, and so it gets reported, well, the mean salary is $112,000, and everyone gets upset. Oh, that's too much money to pay them. And actually, most people make this much money, but because it's such a skewed distribution, you only need a few people making this much money to really skew the mean. All right, enough of my soapbox. Moving on. Are there questions about this and how to visualize mean, median, and mode? Get a drink of water here. Any questions? Okay, I'm watching the chat box. Now, here's what spread means. Okay, variance and standard deviation measure precision. That is precision of the data points that were collected or observed. Precision means how close are the data points to each other. Okay, now it makes sense that both variance and standard deviation speak to precision, because remember, standard deviation is just the square root of variance, or variance is just standard deviation squared. Same thing. Okay, so depending on if your values are precise or not, we can visualize this on a dartboard. So here, let's start here in the upper right-hand corner. These black dots are all your data points that you measured in the lab, and you'll see that they're tightly clustered. So this particular group of data points have very high precision. There's very little variation. There's very little distances between the data points. Okay, and remember how you measured the distance? You took the mean of those data points and you subtracted the actual values to each data point. So if they're tightly clustered, those distances have to be small by definition. Okay, all right. This graph or figure, these data points are not very precise. They're all over the board. So you can imagine that these standard deviation will be quite large. Okay, here's something else. These are a little more precise than this one, not as precise as the one in the upper right-hand corner. And these, again, are very precise data points. Now, variance and standard deviation. This is a technical statistical perspective called precision. Okay? When you hear the term accuracy, that implies the observations to the real or true value. So accuracy measures how close your data points are to the real or true value. Okay, so they are two different things. Even though we speak of them sloppily, we often refer to the same thing. Okay, so accuracy, let's assume that accuracy is bullseye. So that's what the true value is. These are not very accurate, right? Because they're not even close to the bullseye. However, they are very precise. These are both accurate and precise. These tend to have low precision and overall low accuracy, although the mean is probably very close, right? The mean is probably over here where my cursor is. And these tend to have better accuracy, but also low precision. So the point is this: precision is just the relationship of the data points to each other. How spread out are they? Accuracy is a term that describes the distance of the data points to the true value, Okay, they measure different things. And as you see from this figure, you can have different combinations, all right, of precision and accuracy. All right, questions before I move on? Only 2 more slides. Okay, give Christine a chance here to type while I take a sip of water. No questions. Okay, trick me. Here we go, 2 more slides. Now, what is a sample? This is an important concept that I don't think your appendix explains well. There is a difference in statistics between a sample and a population, okay? And here's the quick and dirty. The population is a technical term that means all the members, all the data points, right? A sample is a subset of those members. Now, literally the members could be people. The members could be squirrels. The members could be sponges. The members could be corn plants from a field, okay? The members could be anything. We're not talking about people necessarily, even though we tend to refer to populations as people or animals or stuff like this. The member population just means all the members, all the elements. A sample is a subset of that. All right, so let me just, ideally, if you collected or you measured every single member in the population, you would get a very accurate assessment of the true value. You know it's the true value because you measured every single element in the population. Okay? You've accounted for all the variation by measuring every single member of the population. This is often impossible to do. All right? If we want to study, I don't know, pick an animal. If we want to study, this is Michigan. If we want to study wolverines that live in Michigan, is it feasible to go out and collect every wolverine that lives in Michigan? It might be fun to, but is it feasible to? Is it practical? The short answer is no. Why? Because it takes too much time to collect every wolverine in Michigan. It takes too much money. Remember, your experimental design in reality is going to be restricted by both. And so therefore you take a subset of the members, and the whole goal of stats is to measure, that accuracy and precision in the relationship between the sample and the population, because you're hoping that the sample that you took is representative of the population. So let's say we've got one population here, and we've got, 3 different colors? We've got 3 different types of members. We can take a random sample, which is part of your experimental design that many of you are getting at from our work in the last 2 weeks, and we can end up with a subset, a sample that looks like this. Now that's not bad, right? I've got all 3 colors represented, all 3 members represented, and of course, if I take an extra one, I gotta have at least one extra one. So that's not bad. But it's also possible that I could take a sample and I could end up with all the same type. It's also possible that I could end up with not every member represented, okay? And so the differences here between the samples, this is called sampling bias or sampling error. And sampling error doesn't mean that you were out in the field collecting wolverines and you made a mistake, per se. Sampling error means that you were out in the field, you collected a sample, and the error in the sample is not the error because of you. The error in the sample just happens to be the representation of the members that you collected as your sample. Does that make sense? So I don't want you to walk away with thinking sampling error. Oh, I made a mistake, made a mistake. There might have been a mistake in the sampling design, right? That's called sampling bias. It might have been a mistake in the sampling design. That's called sampling bias. So remember when we did our exercises the last 2 weeks and I said, I really want you to make a list of the potential biases that you might have. That's why. Because if you can identify those biases, you can take away those errors. Okay? The sampling error is a technical term in stats. It is not referring to the technician error, your error, or my error. It's referring to the error, the discrepancies that occur when you take a sample from a population, because you cannot collect all the members of a population. Is that clear? Does that seem reasonable? I'm going to pause here for a second before I go on. I'll wait for 1 or 2 people to respond. So I'm asking you if it seems clear so far, that description between sampling bias and sampling error. Okay. All right, good. All right, thank you. Just make sure everyone's still there. Maybe I lost connection. Okay. So your real goal as a scientist is to minimize the sampling bias and to minimize the sampling error. So let me ask you a question and I'll wait for you to respond. You see what's on the screen. How can I minimize my sampling error? Or how can I improve the accuracy from what you see on the screen? What things can I do? Bingo. So Christine has a great point. So one thing you can do is collect more samples. Perfect. That is probably the easiest way to improve your accuracy. Okay? Measure a larger sample. Take more members from the population. Okay? Because by taking each member, the sample becomes more and more like the mother population. Right? As you put more dots over here, the sample then becomes to look more and more like this. Okay? So that's called increasing your sample size. Perfect. Anybody else with another idea? Yes, Leah. Am I saying it correctly, Leah or Leah? I'll go with Leah until you correct me. Okay. You want to pick the members at random. That is true. Okay. This is called sampling bias, right? So you want to remove the bias and pick the members at random. So why? Okay. So here's an example. It doesn't have to do with biology per se, but every day from now until November 3rd or 4th, you're going to hear about an election poll. Okay. And let's say one of the questions in picking a candidate is, do you think that university tuition should be capped and reduced? Okay. And if you want a particular answer that says yes, where do you go to ask that question? Who do you ask? Who are the members of your population that you would ask that question to intentionally to get the answer yes? Who do you think would be the best people to choose? The students, exactly. The people paying the tuition, right? So you hang out outside in the parking lot of SVSU and you ask students, do you want your candidate to put a cap on university tuition? Would you like them to fund more of tuition from the state so that you don't have to pay as much? And of course, the answer is going to be yes. And you show the results to the poll, right? That's sampling bias. So going back to Leah's point, pick the members at random. And by the way, I don't necessarily disagree with that point. It should be. All right. There's one more way that we can reduce our sampling error. Anybody want to take another stab at it? So we've got to increase the sample size. This one's a little more subtle, not explained in your appendix well at all. Let's see what Christine's got to write. Mr. Miller, you all right today? McKenna, you all right? Haven't heard from you. All right. Christine says do multiple trials, and that is also correct. All right, good. And in this particular case, if I use my terminology, Christine, I'll say you take more than one sample. Okay. So maybe Luke has a research team with McKenna, and they go out and they collect one sample of wolverines. And then Diva has another research group with Christine and Leah, and they go out to a different part of Michigan and they collect a different sample of wolverines. And so we are making multiple samples, right, from the mother population. The mother population in this case is all wolverines in Michigan. And so each of them will then go out and measure their central tendency and their variation in those data sets and their own data sets. And then we can compare their data sets to each other. Because if they're both accurate, those different estimates should be similar to each other, right? If there's no sampling error or minimal sampling error between these different samples that we took, the variation between them should be relatively small. And this is what standard error measures. Now, this is tricky, right? Standard deviation is a single sample. It is the measure of the spread in a single sample. Okay? Standard error measures the variation among the means. Where did the means come from? The means come from the different samples that you took. Okay? So in this particular case, here's the mother population. 5 samples were taken. Each group measured their own mean, standard deviation, and then we compared the means, the 5 means, into this measure called standard error. The abbreviation is SE, okay? And STDEV is the standard deviation, and then you divide by the square root of n, the number of samples, in this case 5, right? 1, 2, 3, 4, 5. Standard error measures, it's supposed to be variation among the means. Okay, McKenna, I will post the slides when I'm done. Yes, I can. Okay. It'll take me about 30 minutes after we're done, but I'll post them so you'll have them. All right. So I stop there. That's 30 minutes, 35 minutes of stats intro. You've got the 6 pages in that appendix to help you. And of course, you can use any reference that you want on the internet. You can go to Khan Economy to get this re-explained. Of course, you can always ask me. I'm willing to answer them. So I'm going to pause right here. I'm done with the stats introduction. And I want to know, are there any questions, Emily, Courtney, Anna, Amanda, related to stats? What they're measuring? Any of the terminology I can clarify for you? Okay. Is this old hat to you? And meaning, have you heard this before? Is everyone sitting there eating popcorn thinking, when is this guy going to move on? Or am I boring you, in other words? Have you already done this? You've already done this, I go a lot faster. I just don't want to bore you. That's okay. Okay. Now, if you're hanging out on a computer and you got a hand or you got time, maybe you open up an Excel file. I'm going to stop sharing. I'm going to now open an Excel file for you. And I'm going to show you some tricks in Excel, perhaps you do or do not know. And because you're going to be using Excel to do this homework. Okay? Now, can you see the, yes, I'm assuming you can see the screen. All right. Now, I just have some numbers in this Excel file. My layout of the Excel, all the tools at the top and the toolbar and where things are might be a little different on your computer. I work on a Mac. I might have a different version of Excel than you have on your computer. If you have a newer computer, that's okay. The same general principles apply. All right, good. Everyone can see this. So I'm going to just give you some dumb terminology. If you know this, my apologies. My experience is when I see biology majors in their 3rd and 4th years, they still don't know some of this. Okay, so if you know this, have some more popcorn. Okay, so you'll notice that the pages are called sheets in Excel. You can open up the file and have many sheets. All right, at the very bottom, you can see my cursor. This is called sheet number 1. You can double click this, when it turns highlighted, I can rename it. Okay, if I want another sheet, I hit the plus sign, I got a new sheet, and I can rename that. Okay, and you can toggle back and forth and you can see the difference. Okay, now the reason I'm telling you this is because the way I run my data sheets is I'll have one sheet and I'll call the raw data. Okay, and my matrix will be in here. And anytime I want to make a manipulation of the data, I take my raw data. What do? I'm going to make it, you can do, let's see, sorry. I know where the 5 and the 1 is, but that's about it. I can either highlight my data, copy. So I'm using the shortcut to copy. You can also go up here to the screen, edit, copy, edit, paste. But if you learn the short notation, right, it goes a lot faster. I can copy it, go to my new sheet, and I can paste it. And here I'll say means. I'm going to calculate the means on this sheet. So the reason I do this is because if I ever make a mistake, I can always go back to my raw data, which I know I don't manipulate or change, and then I can just cut and paste, or I can copy the sheet. Okay, if I'm on my raw data, I can go to edit, I can go to sheet, I can go to move or copy sheet. I will create a copy, move it to the end, I'll push OK, and there's my raw data too. And I just rename this. This will be modes. All right, whatever you want to do. Now that's just a piece of advice, so that if you take your raw data set, your template, and you start messing in it and you make a mistake, you have to go back online, re-download a new one. Okay, whereas if you just keep it in the sheets, you're good. All right, that's my first piece of advice from experience. Now, you'll notice I have a column here. You can highlight the column by clicking here. You can highlight the rows by clicking here. If I go to this little funky symbol in the corner, I can do it all at once. Okay, now each of these blocks is called a cell. Right? These blocks is called a cell, and the cell has a name. So the cell so here's my formula bar. This is my formula bar at the top. Sometimes there's this funky F, which means function, right? So there's the value that's in the cell. The cell number is over here, D6. So every cell has a name, has a reference, has an address. It's the column, which is always a letter, and the number. All right, so if I go D6 and I go down, that'll be D7, 8, 9. See that? Okay, over here, I have L8. Okay, you probably already know this. I'm just pointing this out for a reason, because when we make a formula, this is what you'll need to be able to reference. All right. If you don't like the way this is set up, you can go here. You can align it to the middle. You can align it to the left, to the right, however your eyes see. We all have our own preferences. That's up to you. And my data matrices that I work with, I like center alignment, because that's how my eyes work. So I'll do that. Okay, that's easier for my eyes to see. Okay, we have some numbers. All right, so the important part here is that at some point you're going to want to be able to calculate something. Okay, so I haven't put any column headings on. Okay, you can put whatever you want. Let's call this test 1. This is test 2. So these are students. Do this. See how it's going to automatically fill it in for me already? If you want, you can come up here to your formula bar, change the 1 to a 2. Now here's a nice trick. I want to label all these to student 20 or whatever it is. Over here in the right-hand corner, there's a solid box. If I put my cursor there and pull down, it automatically fills it in. Okay? I don't have to mess around retyping this every single time. Okay. Likewise, I could have put student here and just made this a 1 and a 2. And if I do the same thing, bingo. All right. So these little tricks can make your life easier. All right. So let's say I want to measure in this column, I want to calculate something. The difference between test 1 and 2. So I need to subtract test 1 from test 2. Now up here in my formula bar, all right, anytime you want to use a function is a calculation in Excel speak. You need to use the equal sign to start the function. Okay? Now, the plus sign is addition, the negative sign is subtraction, the asterisk is multiplication, and the backslash is division. So those 4 binary operations, you're probably just going to learn the which button to push. Plus sign is plus, negative sign is subtract, asterisk is multiplication, the backslash is the division. If you use the wrong backslash, don't worry, it won't work. You'll know to use the other one. So if I want to subtract the 2, there's multiple ways you can do this. I know that test 1 is in L6, so I can type L6, and I want to subtract M6. When you're done, you notice that it highlighted the 2 cells in my equation. Now I'm moving my cursor around, but I'm not going to hit a button. If I hit another cell, it's going to mess the equation up. So if you start doing this, don't click. Okay? I'm going to hit enter, and it's calculated it for me already. Okay? Now, see the square again on the bottom? I can do this here if I want. This will be equal sign L7 minus M7 equals. However, Excel is smart enough that if I drag this all the way down, it will put the same equation in each of these cells. Okay? And you'll notice that it's a relational equation. So look at the equation box up here at the top. That's L and M6, and then that's 7. That's row 8. That's 9, 10, 11, 12, 13, 14, right? So that's called a relational. This is when you drag and drop, it keeps the relation among the cells, which works in some cases. Okay, which will work in some cases. I'm going to show you one more trick. I'm going to copy and paste this. I'm going to post it there. Difference between test 1 and the average. All right, so now we're going to get into some good stuff. I want to know the average of the test, or the mean of the test. So I'll tell you what, let me use mean. So I'm going to put mean there. I'm going to go into, I want the mean to calculate in this cell. Again, there are multiple ways to do this. There's a funky, there's our sigma sign again. You see that? It'll be somewhere on your toolbox. If I click on this arrow, they call it average. I'm going to put average. Now you'll notice in my cell, in my box up here, it's already input the notation. The equal sign means I'm going to calculate something. The average is the stat. And then anything in brackets, or these are parentheses, anything in parentheses will be the cells that will be operated on. In this case, it was smart enough, right? And it selected all of these cells. If you don't want this empty cell to be included, you can adjust the size of it, okay? But I don't care if it's there or not. So I'm going to go enter, and that's my mean. Now again, if I want the mean calculated for this test, I can drag it over, and I calculate the mean for that test. Okay, so these are shortcuts. If you end up writing it out every time, that does not matter. And in fact, it's probably better that you do it until you get the hang of it. I don't know how often you play with Excel, so I'm assuming you've got minimal experience. If I'm wasting your time, please let me know, and I will do something else. Okay? All right. So here are my means that I've calculated, again, with this drag and drop. What is something else we had to calculate? The median. All right, let's calculate the median. So I'm going to come up here. Now you'll notice that the median is not listed, but I can go here to more functions, and I can come down here. Oh, look, there's median. Now, there's several things you can do at this point. It depends how you operate. I can highlight median, and then I can go insert function, and that put the equation in. So now look over here in the equation line, equals median, and now I need to specify what you want me to take the median of. So don't worry. So right now it's highlighting L22, so it's not that smart after all, right? As long as I don't push anything, I want to collect the median of this row, right? I'm going to put my cursor here. I'm going to click the pointer. I'm going to keep the pointer down while I move the cursor. I'm going to let go once I have it highlighted. I let go with my button. I hit enter. We'll see, this is what I mean. You can't hit enter in this particular phase. I got to hit done over here, and there's my median. I can do the same thing, drag and drop. Now I got the median of test number 2. Okay? What's going to happen over time is that you're no longer going to need to use the formula builder. You will just memorize what the notation is, what this prefix is for a particular function. Okay? Over time, you're going to memorize it, and it'll be easier to actually, when you get down, see how I messed it up? I was in the equation box. And so if that happens, that's okay. I'm just going to hit delete, and I can retype median. It does not need to be in capital letters, and hit enter again. Okay? Worst comes to worst, you make a mistake, you delete it in the cell, and you just redo it. Let's pick one more, standard deviation. Go over here. I go over here to my formula builder. I need more functions. Now, it does not show up. So in the search box, I can put standard deviation, hit enter. Doesn't like me. Let's do this. Close it out. Do it again. Oh, if I can spell. Still doesn't like me because I can't spell. I saw you. Standard deviation. I'm going to insert the function. Again, it's not in the right place. I'm going to highlight it. Once I've got it highlighted, I hit enter. I can't hit enter. I hit done. And there we go. And I'll drag and drop. Okay, you with me so far? Just some tricks. The only way you learn this is to make mistakes. And the only way you make mistakes is just to sit down and play with stuff. All right? The more you do this, the easier it gets. The easier it gets, the less intimidating it is, the less frustrated you're going to find. All right. Now, before I put a formula in over here, I want to make one comment. What do you notice about all of these data points that I have highlighted? They have different numbers of significant digits, right? These values have two significant digits. This has however many, and that has however, right? That's how they want to calculate your GPA to 12 decimal places. Who cares? It's irrelevant. So one thing that's important in science is going to be you must have a constant number of significant digits. Now, your appendix does not discuss this, right? But the short version is you can go to one significant place past 45. So in other words, I could have 3. I could have a decimal, 45.2, 45.8. But I want them all to be the same. So I'm going to highlight them all. And you see these two little funky icons here? One of these will make fewer significant digits, and one of them will increase the decimal place. So if I want them all to look the same, I can go here and keep going down until I have one decimal place. And now that is acceptable. Okay? And you'll notice if I click here on the standard deviation, look, I should get 2.7. But if I make it bigger, it's going to round the 24, 2.7, 2.71. Okay? Just let Excel do the work for you. Okay? All right. Show you one more thing. We'll add a number over here. So the difference between test 1 and average, I'm going to do this a different way. I'll put equal sign. I'm going to go over here. I got my cursor. I'm going to go, here's test 1. I'm going to click the cell rather than type it, minus sign, and here's my average. I'm clicking it, and you notice it filled it in that way, and then I hit enter, and there's the difference. I'm going to drag and drop. Okay, the difference between test one and average, hey, remember that standard deviation equation I just gave you? The sum of squares. All right, well, here's the difference. And if I want to square this, I'm just showing you different functions real quick because you're going to have to do this for your homework. Square the difference. I can come down here. There is no square sign, so I'm just going to take this. I'm going to multiply it by that again, and that's my square. I'm going to come down. I don't like all those numbers. I'll come over here. I'll go to 1 decimal place. Now, I'm going to put sum. Now I'm going to add them. Sum. There's my sum of squares. So I just calculated the own standard deviation using what is called the function, because here's the function. It says STDEV, and here I can calculate standard deviation by my hand. Oh, wait a minute. I need to know the count. I need to know my sample size. Right? So the sample size is called count. So it will tell me there are 15 values, right? But I need to know the square root of n. So this is going to be back up here, more functions. There's, why does it disappear on me? Square root, SQRT. I don't know why it doesn't like me today. Here, so I'll do this a long way. I'll equal SQRT, parenthesis, put that in there, hit enter. Don't like all those digits. Okay, and so I can calculate, or I should be able to calculate standard deviation, which is what? Equal to sum of squares divided by the square root of that. I'm way off, so I made a mistake somewhere in my math. Oops. The sum, what did I miss? Oh, yeah, doesn't matter. I calculated the variance of two different things. But anyway, you can calculate it either using the function or you can do the long way. And there's nothing wrong with doing it what I call the long way, where you do the individual steps. You'll actually learn more doing the individual steps, and your homework asks you to do both of these. Okay? I will do one more thing, and then I'll open this up for questions. And that is, let's say, so, oh, that's what the problem is. So you see this difference between test one and average? So look over here at my equation. Test one is L6, and the average was L22. But when I just dragged and dropped, it's now telling me that the test difference is L7 and L23, but L23 is the median. What does this one say? Well, this one says L24. I don't want L24 either. So this is called this is that relationship amongst the cells. Sometimes you want the same cell to be used for every calculation. And then in this case, the same cell to be used in every calculation is the mean. All right? I want that yellow cell to be present in every equation. So I could take this. I could, is that going to work? I could, no. And so you come up here into your equation, and the dollar sign fixes the column, and the dollar sign fixes the row. Now, this is a trick. You have to put the dollar sign in front of the column and in front of the row. You can't just put it in front of the column or in front of the row. You got to do both. I'll hit enter. And now when I drag and drop, watch the numbers change. All the numbers changed. Okay? And so that is the true difference between the test one and the average. Okay? All right. So there's just a few tricks. This is just playing with stuff. Of course, you can always highlight things, pick it up, move it around. You can always take this, you can move it there, whatever you want to do. If you make a mistake and you try to load them on top of each other, it's going to tell you, do you want to really replace data points? Okay. All right. So I'm done. We've had 35 minutes on basic stats. We've had about 25 minutes on how you might get around Excel. And so your homework is already listed, it's already posted. Your homework, I've given you 2 Excel files. They're called tutorials, tutorial 1 and tutorial 2. And it's really to get you to start playing with this so you can make some basic calculations. Because what's going to follow is using Excel to make some graphs. But let's do this first. Okay? At the end of the week, by Friday, there are 11 questions for you to answer and input your results from your calculations of these 2 tutorial works, these 2 2 tutorial Excel files. Okay? So in other words, follow the directions, open up the tutorials, go step by step across the sheets. So at the bottom, that's how you manipulate it. There's about 6 different sheets in each tutorial. The last sheet is called exercises. That's the one you're going to do. You're going to keep your results handy, and then you're going to input those results into an assignment in Canvas by Friday. I think Friday at 8 o'clock, Friday at 5 o'clock. Okay, and that's all we're going to, I say that's all we're going to do, but that's going to keep you busy this week, I think. And then we'll add something else. We'll add another layer, etc. So there are questions for me at this point, either with the stats or with the Excel or with your homework. Please ask. It's easier to do it now with everyone here. So I guarantee you somebody else has got the same question. Yes. So Diva points out that for the homework assignments, do you want the number of significant figures to be the same as the data sets they give us? Yes. And in fact, I've already put in the answers, and the answers assume the correct number of significant digits. Okay, which I think is 1 decimal point. We can talk about significant digits next time and how to calculate, how to determine the number of significant digits if you've forgotten. Okay, but I think the answers require 1 decimal place. Okay, does that answer your question? Anybody else with a question? Okay. Any other concerns we're having? This is week 4. Are we settling into a routine? You feeling okay with your other courses? Anything I can help you with, navigate? I'll ask you one more question then, and that is, do you think we're going too slowly? And I can give you, we can cover more each week. Is this about the right pace? We're going too fast? Because I'm willing to adjust as you need. Okay. All right. Fair enough. All right, I'll make you the promise. If we get to the point where we're going too slowly, please stand up and say something so I don't waste your time. Okay? My approach is that I'm okay doing these chunks that we're doing. Okay? And as long as we're doing them and we're learning them and doing them well. So you don't have to come back and relearn it. Okay? All right. I'm done. I'm going to post these slides, 6 PowerPoint slides, here in a few minutes. Otherwise, you got 2 hours left in your lab time to get started on your tutorial sheets. there's no questions, you're not voluntary if you want to stay and ask questions. Otherwise, have a great week. I think it's going to be a little sunny this week, so you might as well enjoy it, because you know it ain't going to last long. No worries, Sarah. I think I will open up a discussion that's just called Excel tricks or something like this. If you've got a question about Excel and you want to post it there, that's great. Maybe we can learn from each other using that. I'm sure you will find tricks and faster things than I know. I have to look at the file, Tiva. First I have to remember where I put it. All right, I'm opening it up. All right. Oh, I see what you mean. Okay, so you can go to, since beak depth is measured to 2 decimal places, you go to 2 decimal places. Okay. It's actually 4 significant digits because there's a 10 in there, for example. So we'll go to 2 decimal places. All right. Any other questions? I probably got a bunch of people that get mad at me now after I said 1, because it'll mark them wrong. Mercy.